All right, so welcome to the breakout session for protein synthesis. So today I'm going to go over this two-step process. Please know that there are, especially in the first step, a lot of similarities to DNA replication. So there's going to be some things that sound very familiar, which should be helpful. There's also going to be some things that are a little bit new, some changes. There are quite a few steps to both processes. So I'm going to encourage you to pause the video as we go, take notes, and then I would really encourage you also to go and use some of the other resources that are available. Watch a follow-up video. There's some online activities where you can actually practice doing this step by step. Really the most useful way to make sure that you understand the stages and what's going on. When we are back together as a group, we'll do some practice together as well. This is going to give you a head start and give you a great foundation to prepare for that roadblock and the next tier. Okay, so quick question, maybe to pause and think about, is the only time we need DNA when we copy it. That's all we do, copy it, send it on to the next cell. I think logically we can all say, well, no, there's no point in copying it if we don't need it for other things, right? So what else do we need it for? DNA really is the code, the instruction manual to make proteins that cells and multicellular organisms like us need to function. Proteins do almost all of the critical jobs within the cells and within organisms' bodies. And everything, things we take for granted. Your hair is made of proteins. The color of your skin is based on the amount of proteins found in your cells. And you could go on with many more examples. So proteins really are critical. The production of those proteins gives you the physical traits and characteristics that we assign to people. We describe when we describe what someone looks like, is really describing the protein content of that person. Okay, so the gene that codes for these proteins is a piece of the DNA. So it's not the entire strand, it's one small piece of that strand that codes for that specific protein. So one gene, one protein is a rule in biology that each gene codes for a single protein, and then that protein helps to determine a trait. Now, please know that sometimes traits that we see are determined by more than one protein interacting with each other. And then some are the result of a single protein. So again, skin color, melanin production. Really that melanin and the amount that you produce leads to lighter or darker skin tones. Whereas there's other proteins that interact together to give traits. For instance, eye color can be a combination. Hair color, not every strand of your hair is identical to the other. So there's some interaction there. All right, so we love rules in biology. One of those huge components of biology is critical is the central dogma. And it's really these steps that we're going to break down into how we go from DNA to the protein that leads to a physical trait. So that central dogma tells us that we take the DNA, we read it, we convert it into a molecule called RNA. Yes, its name is very similar to DNA because they are very similar with some differences. That RNA is going to give instructions on making the protein, and that protein is going to lead to our traits. The reason why we need this RNA step in the middle is DNA can't leave the nucleus. So DNA can't go out in the cell and tell ribosomes, which are the molecule that are making the organelle, that are making the protein, can't tell them what to do. They can't leave the nucleus, so they have to send a messenger. So that's what this RNA really is. Okay, so the two steps of this process, as mentioned in the beginning, are transcription and translation. So before we really dive into the steps, we want to talk a little bit more about RNA. How is RNA different than DNA? They sound so similar. So some of the big differences, RNA uses ribose as its sugar instead of deoxyribose. So we have our nucleotide with our phosphate, our sugar, and our base. The sugar in the RNA is a little bit different than the sugar in the DNA. The other big difference is DNA we know is double-stranded. RNA is single-stranded, so it only has one side because it only has to go out, be read for a short period of time, and then we want it to break down, so it doesn't need to be really strong. And we know our four nitrogen bases are A, G, C, and T in DNA. However, in RNA, instead of using T, they replace it with uracil, so a U. So there is a difference there. There's a base that's different. It's still going to follow T's base pairing rule. So if there is an A in the DNA, instead of having a T put on the RNA, you're going to put a U there, and you'll see that coming up. There's more than one type of RNA. The biggest one that we're going to talk about, especially in this first step, is messenger RNA. And its job is exactly what it sounds. It is a messenger. It's the one that's going to take the code from the DNA. It's going to make a copy of that code. It is the copy. It's going to leave the nucleus, go out to the ribosome, and give it the instructions. So it's taking the message from the DNA to the ribosomes, telling it what to do. Another key player a little bit later is transfer RNA. And again, we're lucky. Scientists were very kind when they named these molecules because their job is in the name. The transfer RNA carries, it moves amino acids, the building blocks of that protein. So as we're building it, and it's made of amino acids, we got to get those amino acids carried to the ribosome so that it can go into the protein, and that transfer RNA carries that amino acid in. One we're really not going to focus on today, but we want to kind of honor that it is another type of RNA, is ribosomal RNA. It really makes up the ribosome. So we say ribosome, and we talk about what it does. Well, it's made up of RNA. So there is RNA within the ribosome. And we know from earlier in the year, we can find ribosomes either attached to the endoplasmic reticulum or free floating around in the cytoplasm. And that does make a difference to where their proteins are going to go in the cell. All right, so we're going to kind of walk through transcription. As you're looking at this, it's going to look really familiar because it really is just like DNA replication. So just as in those steps, in transcription, we've got to unwind, we've got to open up the DNA. Helicase is going to do that, just like in DNA replication. Except it's not going to go down the entire length of the DNA molecule. It's only going to open up a section. You can see this here, this bubble, where the code is found that we need to make a copy of for the protein that needs to be made. You might be thinking to yourself, well, how does it know which protein needs to be made? That's coming from a signal, a need from within the organism or within the cell. So there are signals that are triggering the production. Think of like flipping a light switch and the light comes on. Triggering the production of the protein that's needed. So it's going to trigger which section of the DNA that protein's code is found in order to make that copy for the messenger RNA. So in transcription, RNA polymerase is going to read the DNA to make messenger RNA. So just like in DNA replication, where we had DNA polymerase making the new complementary side, RNA polymerase is going to come in and make the messenger RNA, reading the DNA, and it follows the same rules. It builds 5 to 3, you can see that here, and reads 3 to 5. Okay, so following the same rules that we talked about before. Once that's created, messenger RNA is going to travel out of the nucleus to the ribosome in order to allow for the next step. Now, just want to kind of asterisk here. This is an oversimplification, right? There's a lot more going on than this. You can go out and watch videos that are going to show, how do we trigger this? How do we know when to stop? So there is a lot more at work here. It is a little bit more simple than it really is. But for our purposes right now, it's the foundation. It's what we need to know. Okay, so that messenger RNA is going to head out of the nucleus and it's going to go to the ribosome. Okay, there is some processing that occurs in the nucleus. There's some modification. We got to tag it so that it knows which ribosome to pair up with. So again, a little bit of an oversimplification. There are some other things going on. If you're like, ooh, I wonder what that is. Great idea to join AP Bio because we do kind of jump into that in a lot more depth and you can maybe go in deeper or go in deeper on your own. But for now, just knowing that, there is some modification that goes out in the nucleus just to make sure that that messenger RNA gets where it needs to go and has the right code is about all you really need to know in terms of what happens after the messenger RNA is made. Okay, so quick little review. I'd like you to pause the video, see if you can recap what we just went over by doing a Venn diagram comparing RNA and DNA, and then DNA replication and transcription. So I'd pause it now, try to do those Venn diagrams, compare and contrast, and then you can go ahead and restart the video and we'll go over the answers. All right, DNA versus RNA. What are some of these big differences? So the sugar was one of the differences that we mentioned, that DNA is double-stranded and RNA is single-stranded. RNA is only coding for a single gene, whereas DNA has thousands of genes in this one strand of DNA. Some of the bases are the same. We use A, G, and C in both universally. However, the big difference in DNA versus RNA is that DNA uses thymine and RNA uses uracil. We do have the phosphate component. That is not changed. These are covalently bonded together out here in the backbone. So some of those basic components haven't changed either. Okay, transcription versus DNA replication. So you've got some of these differences up here. So in DNA replication, we copy both sides of the DNA molecule to make two strands, whereas in transcription, we're only copying from one DNA and we're only again copying a gene. We're not copying the entire DNA strand. We use DNA polymerase in replication and RNA polymerase in transcription. DNA replication we only do when we have to go through cell division, and then transcription's happening constantly as we need proteins, which is pretty much every second. In terms of similarities, DNA is the template for both of these processes. They're going to follow base pairing rules, again, except for the UT difference. And helicase is going to be used to open up the DNA in order for this process to occur. So that's a little recap. We're going to move on to translation now. So what happens to that messenger RNA molecule when it reaches the ribosome? Okay. One thing to consider is that this universal genetic code that we've been talking about that we're reading is literally universal. All organisms, from the simplest bacteria to the most complex organism like us, uses the same DNA structure, the same bases, forming proteins, and even some of these proteins are exactly the same or nearly identical. So consider, what does that tell us about evolution, about our relationship with all the other living organisms on the planet? Something to consider, something we'll come back to more at the end of the year. Some things we kind of talked about at the beginning, so we really kind of bookend the year, but want to keep bringing up this idea, these similar characteristics between all living things. And this process of translation is one of those as well. So translation, we're going to make a protein by reading that messenger RNA code. And that messenger RNA code is read not letter by letter, but is read as kind of quote unquote words, codons. Those codons are three nucleotides, three bases read together. So it's a three-letter word for that codon. Those three letters are going to match up with a code on the transfer RNA that's bringing in an amino acid, and they'll match together, and you'll see this in just a little bit. And when they match correctly, it's going to drop off the amino acid that it's carrying. And then eventually all those amino acids come together to make the protein that we need. So here's an example of just a small piece of a messenger RNA code. And here's how we're reading it. Again, three letters is a codon. This first one, AUG, is the start codon. There's only one start. So when we're creating the protein, we don't start until we get to the start codon. Again, nice, easy name that lets us know what's going on. So AUG is the beginning. That's going to put down an amino acid. That amino acid is called methionine, and that's always the first amino acid in every protein. And then we go on to read the next three letters. These three letters code for an amino acid called threonine, and that would be the next one bound, and we would keep going. Again, reading three letters at a time. You might be wondering, well, how do you know? How do you know AUG together is a start? How do you know ACU is threonine? Well, of course, we have a cheat sheet. Okay, so we've got these ones that we know. Again, AUG is our start. There are 3 different stops, and we find these all on an amino acid chart that looks like this. So how you read this one, and they come in different versions. The first letter in the codon is found here. So if the first letter in my codon is an A, I'm going to start here. I know somewhere in this row is the 3 letters that I'm looking for. And then if I come up here and I know what my second letter is, U, right? I know it's going to be here in this box where the A and the U match up. So here I'm looking for AUG, methionine. That's my start codon. As we look through this, we can find our stop codons up here. So there's 3 of them again. These are not things that you need to memorize. Anytime you need to do this, you would have this chart that would give you the information you need. You just have to be able to find the right amino acid. Now these amino acids on here are abbreviated, right? So the name of the amino acid is an LEU. It's leucine, but it's abbreviated here just for space sake. All right, so here's an image of the entire process overall. So we've got the ribosome up here where this is happening. Ribosome has a small piece and a large piece. And then in this ribosome, here's our messenger RNA that's traveling through the ribosome. So it entered here on the left of the ribosome and is traveling left to right across and is being read. So you can see there's kind of 3 spots here. This A spot is where it started. So there was an AUG there where it began here, and then it connected with reading these 3. So you're going to notice these little T-looking molecules are transfer RNA. It comes in with its anti-codon. This is how it knows how to match up. Again, we're still following base pairing rules. So if I have an AAA here, the complement to that is UUU. So it binds, it says, yep, this is a good match. And then it's going to take the amino acid that it's carrying, bind it to the next amino acid in the line. This one is valine, was the last one that was put down. So this one's going to bind to this one. And then what's going to happen is everything's going to slide over. So this one will get kicked out like they're showing here. This one that's currently here in the middle will slide over. And the one in the A spot will slide over holding the entire molecule, and it will continue to grow. And this happens really, really quickly. One reason why I really recommend you to go watch some of the videos of this happening is to see it in action. It's a process, it's step by step. To watch it occur helps it to make more sense. So a still picture can only kind of show you so much. So watching it in action really does help. All right, so this is kind of just breaking it down step by step. The ribosome complex forms around the messenger RNA. We find that start codon and the transfer RNA holding methionine matches up to that. And then we continue to slide down the chain. Think of this as an assembly line, reading 3 letters, 3 letters, 3 letters at a time as a codon, putting in those amino acids that match up. Eventually, we're going to reach a stop codon. A stop codon doesn't hold amino acid. What it does is literally just when you reach that, break the entire complex apart. We stop building the protein, it goes off to do its job, the ribosome falls apart, the transfer RNAs are all gone. The messenger RNA can either be read again if we need more of that protein, or it can be destroyed and the materials can be reused. So here's a great chance to practice. What I do is I have a code here. Again, I would pause the video. Here's my DNA code. Can you write what the messenger RNA code would be? And then what the amino acid sequence, what would the amino acids be? Now, there is an amino acid chart in your textbook, so you could use that one to help you with this. So this is a great spot to go ahead and pause, try to do this on your own, and then play the video and see if you get the right answer. All right, so hopefully you've got your practice done and you've pressed play again. So here are the answers. So here's the correct messenger RNA sequence. So you can see here I had a G, it's going to match up with a C. C, I've got G. A, I've got, oh, if you wrote a T, remember you're thinking DNA. Here it's messenger RNA, I'm going to put in a U. T, I've got A, and so on and so forth. Looking at this right away, I can see if I'm going to start reading it to make a protein, I'm not going to start here, okay, because that's not AUG, but here it is. So I can go ahead and start here with AUG, and that's where I'm actually going to start reading my messenger RNA when I move on to translation. So the first thing we're going to do when we do this, we'll start here at AUG. We're going to read methionine, and then arginine, leucine, proline, glycine, and then I think it's isoleucine. Sorry, I'm kind of cutting it off. And then we would get a stop. And again, you don't write the word stop, you just literally do what it says, you stop. Okay, so I'm also going to attach this PowerPoint to Classroom today. There's some simulations on here. There's a good quiz, practice quiz before you take the actual roadblock. So these would be wonderful tools to kind of review, make sure you feel comfortable, because again, there was a lot going on. Email me with questions if you have any, or email me when you feel ready to take the roadblock, and I will send it to you.